Good afternoon, everybody. Today we are doing the concluding part of apoptosis. We did BCL2 members, caspase, and today we wind up with the two pathways, intrinsic and extrinsic. On 5th August we have the 5th series, in that the questions from apoptosis comes. After this, I will start the biotic factors. Because I am blending the UG and PG, it is not required if you are in BSc 1, 2 or 3, you can't read the topics which are in MSc. If you are getting the knowledge, today time is that you are indoors, you have a lot of time, grab it. Which antibody is present in tears, saliva, it is not reasonable that I am in BSc 1, I can't read this. This is not the possible. Just who are in the BSc 1 and 2, they are right position to start for the IAS. Go for the bingo. Who are in 2nd, 3rd, they are on the brink, they have to start from the today itself. Just to see the videos. I want you all to clear the competitive exams. I want you all do which I did the mistakes, you don't repeat those. Just to put your foot on the accelerator, and through all the mock tests I am giving, when you appear in the competitive, then you will remember such an easy cakewalk for you. So stay indoor, stay safe. I will be right back with the part 2. Bye bye. Thank you. Welcome back. Today we are doing the concluding part of apoptosis. Before starting that, I just want to revise. First, what are caspases? These are cysteine-dependent aspartate directed protease. CA from here, and SP from here, and ASs from the enzyme. Apoptotic caspases are subcategorized into initiator 2, 8, 9, 10, and initiator further activate the executioner caspases, caspase 3, 6, 7. So this we did in the last lecture. Now BCL2 family. BCL2 family is the most notable for the regulation of apoptosis, a form of programmed cell death. And BCL2, they have 3 categories. First is the anti-apoptotic, that is BCL2 and BCLXL. Anti-apoptotic BCL2 proteins inhibited this apoptosis and ensure cell survival. And the second is the pro-apoptotic, that is divided into the multi-domain, whereas in this anti-apoptotic we have the 1, 2, 3 and 4 domains, whereas in multi-domain, that is back and backs, we have 3 domains, 1, 2 and 3, and the 4th one is absent. It is also multi-domain. And BH3, which consists of BID, BAD, NOXA, PUMA, BIM, it has only 1 domain, small, inhibit anti-apoptotic BCL2 member and stimulate by activating pro-apoptotic. So then I want to tell you what we did last time. This is the mitochondria, and this just I told you these are the anti-apoptotic, that is BCL2 and BXCLX, right? And these are basically the back and backs multi-domain, which are called pro-apoptotic multi-domain, and they cause the pores in the mitochondria and release cytochrome C. BCL2 and BCLX, they basically catch hold of back or backs and does not allow the formation of pores and release of cytochrome C, because I told you, and this cytochrome C which is released in cytosol they combined with APOPTOS-1 and procaspase 9 to form a complex called apoptosome, which change the procaspase into the active caspase, and this active caspase changes the procaspase 6 to active 6, and then lamin is cleaved, and ultimately which result in the nuclear fragmentation, which is the one part of the apoptosis which we did right in the beginning. Now comes into the scene the single domain BH3, NOVA, NOXA, BMX, BID, BAD, PIM, right? What it did, it neutralized BCL2 and BCLX, and they are caught hold of it, and which makes the back and backs free, and they results in the formation of pores as they inserted into the mitochondria and release cytochrome C and results in the formation of a apoptosome. This which we did right now is the also called intrinsic pathway. Now I am coming to the pathways. The first pathway is the intrinsic pathway. The intrinsic pathway of apoptosis, that is the death inducing stimuli, are originated inside, that is called intrinsic, inside the target cells itself. The intrinsic pathway mainly triggers apoptosis in response to the internal stimuli, and these are either in biochemical stress, either in form of DNA damage, and this DNA damage we did do in the mitotic cycle checkpoints in short a while, activates p53 gene which inhibit the cell cycle and initiates cell, that is DNA repair. If the repair attempt is unsuccessful, apoptosis can be induced. This is the role of p53. It is a basically we do in the cell checkpoints and also in the cancer. Then the lack of growth factors. BCL2 family we did with the explanation, and here the BH3 which promote apoptosis is it promote apoptosis by inhibiting the anti-apoptotic BCL2 member, this one BH3. It combines or neutralizes the BCL2 members, that is BCL2 and BCLXL. In both the cases, the BH3 only proteins are the key determinant of apoptotic cell death. In normal and healthy cell, the level of BH3 is at low concentration. If an apoptosis inducing stimulus evoked in the cell, the levels of BH3 increases, and that caught hold of BCL2 and BXL. And this increase in level of BH3 only protein cause imbalance in the pro-apoptotic and anti-apoptotic factor. A pro-apoptotic factor called BAX is translocated from cytosol to the outer mitochondrial membrane and gets inserted into the outer mitochondrial membrane. When this imbalance will occur, it will catch hold of it, and these BAX or BAC, it enters the mitochondrial membrane, outer and releases the cytochrome C. BAX proteins create spores in the outer membrane, which increases permeability of outer mitochondrial membrane. Through these pores, cytochrome C is released out of the mitochondria to the cytosol. The level of mitochondrial membrane permeability is accelerated by the increased level of calcium, this question will come, in the cytoplasm, which are released by the endoplasmic reticulum. During times of cellular stress, mitochondrial cytochrome C is released in cytosol, and it binds with the adapter protein APOPTOS-1, which recruits initiator capsase 9 by card-card interaction, because I told you there are 3 points C card and where there is a WD40 repeats which cause auto-inhibitory of the APOPTOS. Please refer to the last lecture. This leads to the formation of capsid activating multi-protein complex called apoptosomes. This I told you, this cytochrome C, APOPH and pro-caspase 9, it results in the apoptosome. This stimulates caspase 9, which in turn activates the effector capsid, I told you. That is, the mitochondria also release a protein called SMAC and OMI into the cytosol simultaneously. Both indirectly promote the apoptosis by blocking the effects of group of anti-apoptotic proteins called inhibitors of apoptosis, that is IAPS. This we did yesterday. Here is the human apoptotic stimuli. This SMAC and OMI are released, and this process we did procapsid 9 to 3, then apoptosis, and this they inhibit the IAPs. IAPs inhibits the procaspase 9 and 3 and it stops the apoptosis, and these SMAC and OMI they inhibit the IAPs and allow the procapsid 9 to activate the procapsid 3 downstream. The anti-apoptotic protein BCL2 and BCL-XL inhibit cytochrome C release and make mitochondria pores impermeable. If BH3 is not available, this will prevent the release of cytochrome C. If pores are even made by the back, first it catch hold of back, otherwise it will again make mitochondria pores impermeable for release of cytochrome C. So now we are coming to the next part, that is the extrinsic pathway. Extrinsic pathway is very important. Before moving to the extrinsic, I will tell you the intrinsic we did DNA damage or P53, mitochondria, cytochrome C and OMI is released and SMAC is released and it initiate 9. 9 will initiate the effector 3 and it results in apoptosis. This pathway is finished. So now we are coming to the next, that is the effector, right? So before I move to the extrinsic pathway, I just want to explain you the intrinsic pathway again with these slides. This is the mitochondria. Here in these mitochondria, the back and back scale, and it results in the pores, and these pores the cytochrome C is released, which is combined with the adapter protein in cytosol and procapsid 9, and they form a complex called apoptosome, and this result in the activation of procaspase 9, and that result in ultimately to the 3, and then is the apoptosis or cell death. Here is the second I told you, same slide, cytochrome C is released and here it come to the cytosol, and here is APOPH 1. Yesterday I told you, last time, activation of APOPH 1, the cytochrome C and hydrolysis of DATP to DADP. And here is a card domain. It is assembly of apoptosome triggered by the release of DADP in the exchange of DATP occurring at the card domain. And there is a 1C terminal is also there, where we have WD-40 repeats which cause auto-inhibitory. And this combined with the card domain, we have the procaspase 9 attached to the card domain. This card domain, see, procaspase 9 is arranged here, and it forms a structure called apoptosome. And this apoptosomes activates the procaspase 9 and then procaspase 3 and then results in apoptosis. See, I already told you with the back and backs And BH3 members, Noxa and Puma, they inhibit the BCL2 members and allow the back and backs to go to the mitochondria, release the cytochrome C when it combined with the APOF. And to APOF, there is a card domain attached the caspase 9 and form the apoptosis, and this apoptosis changes the caspase 9, pro-caspase 9 to caspase 9, then to 3, that is the downstream, and results in the apoptosis. Right? So we can make it more clear. Here I already told you the different like stress, DNA damage, then we have other factors like biochemical stress, DNA damage, and lack of growth factors. Here same it is the back and backs, it releases the DNA damage, back and backs will come BCL2 members and it releases the cytochrome C, and that cytochrome C combined with the APOF1 and pro-caspase 9 to the card domain of APOX1, and they form apoptosis, which activate the caspase 9, which further downstream activate the pro-caspase to caspase 3 and result in the apoptosis. So this is basically the mitochondrial mediated program cell death way. When we go in the IAS level, the question will come to explain the mitochondrial mediated program cell death pathway. It is called intrinsic pathway. So now we are moving to the extrinsic pathway. Here I will explain you just a moment, but first see this slide. Here the factors which is called TNF come from outside. That is why it is called extrinsic, plasma membrane outside. In plasma membrane are present TNF receptors which receive it. And after receiving it, there is a 70 amino acids death domain which undergo conformational changes and changes into the blue colored TRAD and FAD. And this TRAD and FAD, it combines with the pro-caspase 8 to form a multiple complex, and this changes into the caspase 8, then to pro-caspase, which caspase 8 changes pro-caspase to caspase 3 and other result in the apoptosis. So now before moving to next, I will just want you to do the theory part. That is the extrinsic pathway. The extrinsic pathway triggers apoptosis in response to the external stimuli, namely by the special ligands, just I told you, that is the TNF, binding to the death receptors on cell surface. These receptors are typically members of tumor necrosis factors, TNF receptor gene family, such as TNFR1 or FAS. Binding at these receptors leads to the downstream caspase activation. TNF first binds to the receptor, just I show you the slide, that is TNFRI, that is tumor necrosis factor receptor 1, present on the plasma membrane. TNFRI is a member of death receptor family protein that turn on the apoptotic cell death process in eukaryotic cells. TNFRI is a transmembrane receptor with an external ligand binding domain and cytosolic domain. External, I just told you, external domain, it is react with the TNF, and cytosolic we have the death domain and there is TRAD and FAD. The cytosolic domain of each TNFRI subunit contains a segment about, I just explained you, 70 amino acid death domain. Binding of TNF to the TNFR receptor, it causes conformation changes in the death domain, and the two cytosolic adapter proteins TRAD and FAD, blue color and orange color, I just explained you in the slide, and pro-caspase 9 residues binds and form a multiple protein complex. See here, TNF comes and it is received by the TNF receptor, which is outer, whereas toward the cytosol, the other, this is 70 amino acid death domain, and when it reacts, there is a conformation changes occur, it forms two adapter protein, that is this blue color TRAD and violet color FAD. and it combines with the caspase-9 to form a structure that is complex called multiple protein complex, right? So now procaspase 8 and FAD possess a homologous region called death effector domain. The death effector domains of both procaspase 8 and FAD interacts with each other and cleave each other to generate an active caspase 8. This is very important. The procaspase 8 and FAD, they possess homologous region called death effector domain, and they cleave each other and produce the active caspase 8. Hence it contains 4 polypeptide segments which derive from 2 procaspases. Activated caspase 8 is an initiator caspase in the extrinsic pathway. They activate the downstream caspase 3, whereas in case of just we did the intrinsic, where we have basically the not the cytochrome 3 is there, but here we have apoptosome, and then this apoptosome we have the that is active caspase 3, but here it is the active caspase 8, whereas in case of intrinsic it is caspase 9. So one is reduced here, and they activate the downstream caspase 3. They carry out self-destruction and apoptosis of the cell. Another commonly observed extrinsic pathway of apoptosis is in human is the killer lymphocyte. Very important. Very important. Through Fas ligand and Fas protein by mechanism. As shown, the killer lymphocytes can induce apoptosis by inducing a protein called Fas ligand. The Fas ligand binds to its receptors called Fas on the plasma membrane of the target cell. Similar to the death domain of TNFR1, the Fas protein can recruit the intracellular adapter protein, which can aggregate procaspase 8 molecules. The procaspase 8 molecules are then activated to caspase 8, and that is in turn activate the downstream executioner caspase 3 to induce apoptosis. So this is the extrinsic pathway. Here we have the death ligands, then death receptors, this is TNFR1, whereas in case of killer lymphocytes it is Fas ligand, and then it initiate caspase 9, which initiate the effector caspase 3 downstream and result in the cell death. So this we did just now. Now we are coming to the next, that is the killer lymphocyte. I already told you these are killer lymphocyte, and here instead of we have tuber necrosis factor 1, we have the Fas ligand, and these Fas ligand they react with the Fas death receptors, and we have the FAD adapter protein, which is death domain and death effector domain, and it results in the procaspase 9, and which result in downstream activation of procaspase 3, which result in apoptosis. See, same slide, but with detail. Here we have the Fas receptors, then we have FAD 8 and 3. Here we this is the we did just now death domain, we did killer lymphocyte. Here we both are there. One is this one which we did right now, Fas receptor death domain 8 and 3, and this is which we did earlier. Here is TNF receptor 1 death domain, that is TAD and FAD, then caspase 8 and caspase 3. So this we did earlier also, this one. So I want to explain again. In this case, the extrinsic pathway, the receptors are typically members of tuber necrosis factor, TNFR gene family, such as TNFR1 or Fas bonding at these receptors leads downstream caspase activation, that is 9 in case of intrinsic pathway, 8 in case of extrinsic pathway. TNFR1, this one, it is basically a transmembrane receptor. Transmembrane means both sides of membrane with external ligand binding domain and cytosolic domain. The cytosolic domain of each TNFR1 subunit contains a segment of 70 amino acids called death domain. See, binding of TNF to TNFR1 receptor cause conformational changes in death domain, and two cytosolic adapter proteins, that is TRAD and FAD, that is blue color TRAD and FAD, are formed. And these TRAD and FAD, they with pro-caspase 8 residues binds to form a multi-protein complex, and these pro-caspase 8 and FAD possess a homologous region called death effector domain, and there is a conformational changes in these both death effector domains interacts and cleave each other to form an active caspase 8, and this active caspase downstream 8 is an initiator caspase, and they activate the downstream caspase 3. So this way finishes, and I want to again revise you. These are the BCL2 member, that is anti-apoptotic BCL2 BCL-XL. It is made up of 4 domains, and then we have the pro-apoptotic, that is multi-domain back and backs, which cause the pores in the mitochondria. 3 domains, it also called multi-domain, and then BH3, which is only 1 domain, BID, BAD, NOXA, PUMA, which caught hold of these BCL2 and allow the BCL-X to form a pores. And first last lecture we did in C. elegans, factor is from EGL1, and this basically inhibit the CED9, and CED9 inhibit the CED4. And when this is checked and these are released, and CD3, CD4, which is a type of apoptosome, it activates the CD3 and cause apoptosis. Whereas in human, cytochrome C is released, APOF, it will cytochrome pro-Cas9 to form apoptosome with pro-Cas2 3. The inhibitors are IAPs, which are basically caught hold by the SPAC or OMI. So this is basically the crux of intrinsic pathway. Right? See, this here we have the mitochondria, and in this mitochondria, first back and backs, which are also called anti-apoptotic factor, they enters the membrane and result in the pores and allow the cytochrome C to come out. But they are catch hold of by these anti-apoptotic BCL2 and BCL. Like they caught hold of it and does not allow it to make pores. If somehow you make pores, it can again make it impermeable. Now comes into the picture the BH3, NOXA, PUMA, BAD, BID, and they caught hold of BCL2 and BCL, neutralize them, allow the back and backs to cause a pores, release of cytochrome C, and this cytochrome C, which attach with the APOF1, and APOF1, I told you 3 regions. N region, which is called CORD region, and this CORD region, it attached pro-caspase 9, and they form with the help of DATP, they form a apoptosome. Whereas in C region, there are WD40 repeats, which are auto-inhibitory, make this APOF1 auto-inhibitory. And these apoptosomes in downstream first change the pro-caspase to active 3, and then to 6 to then active 6, then lamin to cleave the nuclear fragmentation. Right? So this is the basically the pathways, extrinsic and intrinsic, we did in detail. So now I finishes it up, the apoptosis, and after this I will take the biotic factors. So please revise these in detail. Here you will see I explained you. This is the Fas ligand, and these are the right. So if you have any problem, please revert back to me, because today we finished And after biotic factors, I will move on to the cell cycle checkpoints, and the maximum questions come from the antibody, this apoptosis, cell cycle checkpoints, cancer, and transcription, which we will take side by side. After checkpoint, I will go to the cancer, because in checkpoints I did the P53, and cancer I required the detail of P53, and after that I go to the transcription part. So then we left with a small topics we will do side by side, plus please revise it, because now we have 60 videos, so you have to do all the families also. If you want to clear IAS, so you should have a good knowledge of modern and traditional botany. So blend is required, whereas in UGC just modern 90% questions will come, but in IAS it is a blend. So please revise. Any problem, revert back to me. Thank you. Take care. Stay indoors. Bye bye.